So let me start. So I'm going to speak about virtual cells and digital twins, AI in personalized medicine. And I would like you to take a look at the image. That's a very famous histopathology image, an H&E stain. And at the first glance, this might be a familiar sight for many because this is an image around for many, somehow almost over a century, and has been one of the cornerstones of kind of clinical oncology. It gives a pathologist a window to understand a tumor's architecture. But in recent years, we've really seen a transformation of tissue imaging, and the world has become much more colorful, right? So here, this is one round of staining, there's another round of staining, and so on and so forth. And we've really reached an age of spatial biology, where we have now a technology here known as spatial proteomics that allows us to measure in one single experiment almost 40 to 50, sometimes even more markers, proteins, and their localization within a tissue. So every different color here is the intensity of a single protein in a location in a tissue, which provides us really like somewhat molecular insights into a patient's tumor tissue. It allows us to understand cell regions, cell types, subcellular up to a subcellular or even like somewhat cell level resolution. And given this somewhat in-depth analysis of a patient's tissue, we can look at very different questions. It allows us to understand really regions of aggressive cell growth in a tumor cell, the understanding and infiltration of the immune system within a tumor. And if we look now at patient cohorts, these technologies really give uncover clinically very relevant patterns. They allow us to really understand cancer and point to a way to very personalized medicines. So what concretely can we do, and what do we really aim to do? These advances, they allow us to, when especially integrated with other diagnostic technologies, to transform how we characterize tumors. They help us classify tumor subtypes, assess cancer stage, and more precisely, they really reveal how features of the tumor microenvironment, such as immune infiltration, cellular architecture, stromal composition, are really linked to tumor evolution, progression, or advanced stages. And then they can link to clinical outcomes, such as overall survival, progression-free survival, and so on and so forth. At the same time, they also enable the discovery of biomarkers, which are, or spatial hallmarks, that allow us to understand or guide prognosis and treatment planning. But the impact is much beyond this clinical translation. It really also provides us an insight into understanding of cancer biology itself. By targeting dozens of markers simultaneously, measuring dozens of proteins simultaneously, we get an idea about cell identity, cell states, subcellular localization, and so on and so forth. We can really map how a cell interacts with each other, how they interact with an extracellular matrix, identify neighborhoods and of spatial enrichment, and even really chart the architecture of a tumor ecosystem as a whole. And all this really brings us into a new world. So we can go beyond histopathology, we can really look at spatial proteomics, spatial transcriptomics, a deep screening of a tumor tissue. We are at a pivotal point where we can now generate an unprecedented wealth of data from every tumor we study. So beyond those spatial methods, of course, we can map this to genome sequencing. We can look at lab tests and lab results, and so on and so forth, and build atlas-like data sets of enormous size where each modality really offers a different lens on the state of disease. And together, they really create a rich, multidimensional view on tumor biology. But of course, with this richness in data comes a very new challenge. How do we really make sense of it all? Like, how can we look at the volume, the complexity, the heterogeneity of those data that very much exceeds everything a human can really look at with just staring on these patterns of colors? We might not really easily extract everything we want to know about the tumor. We would like to understand different patient cohorts, basically compare those complex patterns of different patients, compare them to each other. What has worked for one patient, does it also work for a potentially different cohort, a different patient with a different molecular profile? So AI is really emerging here as one tool that might look through the noise, that really is an essential partner in cancer care, enabling predictions of molecular alterations, therapy response, patient outcomes, and bringing us closer to a personalized medicine. Because what we should not forget is cancer is not just one disease. It is incredibly unique, very unique for every different patient. And this is why, in order to treat cancer effectively, we really need to understand each patient's tumor in all its complexity. And spatial biology, powered with AI, is giving us one tool to just do that. So here I just laid you out all the tasks that we might potentially be interested in. At the same time, we have all those different technologies or diagnostic tools that we are working with. That kind of goes beyond what AI so far has been able to achieve, right? Usually we have always one task, one data input, and we can build very specialized models that really excel in this task. One big example here might be AlphaFold. We are looking at a protein structure, a protein sequence for which we want to predict a structure. So the very core question is, how do we build those models that are kind of task agnostic and that can incorporate this wealth of data, right? Where every data modality is very unique in its own, has very different data types and complexities. So we have a fundamental challenge also on the AI side, where we need to think and rethink the way we design AI systems to handle all those type of challenges. One emerging concept here is this idea of building a foundation model or a generalist model, a model that, given one data modality, can solve many different tasks by finding somewhat universal representations that allow us to tackle, for example, prognosis, diagnostics, understanding basically, or retrieving patients that might be relevant to make decisions on a particular case. And what we've seen in the past year, right, it's not very old, is an emergence of several foundation models, in particular here for digital pathology. So models that really are trained on a massive collection of data sets of histopathology data. And these data now provide a universal representation of the state of the data that then allows to tackle many, many different tasks. This is just for histopathology. But even if we already go to the data technology that I showed you at the very beginning, right, that makes tissue biology so colorful, how do we really deal with this type of data? Usually in computer vision, we always have the same set of inputs, red, green, blue, same 3 channels. I just showed you that we can take pictures of channels, of images, tissue images, with almost 40, 50 channels. Every clinical study is going to look at a different set of proteins that are particularly relevant to this patient, to this cancer, and to the kind of objective of the study. How do we build AI tools that can deal with so heterogeneous data, high-dimensional channels, where every experiment might have a different combination of input channels? So even here, we don't really know how to build those foundation models for spatial proteomics. And then the next step, how do we integrate it all, right? How do we actually build multimodal foundation models that incorporate many different diagnostic data to really make the connections between a genomic profile and a tissue image I have? Because I need to incorporate it all to have a best shot at really understanding the intricacy of this patient's kind of diagnosis or kind of state of disease to make the best decisions and to also learn the most, develop new drugs, find new biomarkers that might be indicators for a particular treatment choice or prognosis. So many questions, and I think the root cause of all these problems is the fact that cell biology is incredibly complex, meaning every time we want to take one measurement, our measurements are usually indirect. I want to understand gene expression. I don't measure gene activity. I actually look at like RNA counts one level downstream. We have huge batch effects. Measurements are very, very noisy and obscure. Sometimes I need to, or like this really leads to the next point, I need to look at different modalities at the same time in order to get a good impression and understanding of what's actually going on. I need to look at the distribution of proteins, the transcriptomic state. I look at imaging modalities, genomic, and so on and so forth. Most of the time, we have this luxury of those modalities being aligned, meaning I have an understanding of this particular tissue, both from a histopathology and a transcriptomic and a proteomic readout. But in most cases, we have non-aligned data From one sample, I have this measurement. From another sample, I have this measurement. And then last but not least, everything is incredibly dynamic. Like while I speak, we have thousands, like millions of interactions that steer everything. And while every process is dynamic, the measurements we take are not dynamic, because if I take a measurement, often I need to destroy a biological sample. If I want to understand how a patient responds to a treatment, how a cell would change upon perturbation with a drug, I can either take a picture of of the cell before, or a picture of the cell afterwards. This entire AI dogma, I have an X and I predict the F of X, and I have aligned pairs, is something we are never provided with, except in a few cases. So we need to rethink the way we design algorithms to really make them work for biological data. What we've seen, though, is a revolution in the omics, and we are all using it every day, right? And this, I would claim, revolution is built on really like many years of specialized architecture design, specialized to the data that we are dealing with. Sequences when we work with text, grids when we work with images. We have modules such as RNN or CNN structures, self-attention, very much tailored to the data that we are working with in computer vision or not natural language processing. We have overall architectures that are tailored to the data structures that we are working with. And we also have learning objectives that we use to guide the model training that are tailored to the data that we are working with. Next token prediction, because our language, right, is one word after the other. Masking, because if I mask a tail of a dog, right, I need to understand how a dog is somewhat like structured. I will probably figure out that it always has a tail. That are stupid anecdotes, but they kind of get you a feeling of why those type of learning objectives might work. For tissue, it's a whole other question. What we've set up in the last year is trying to rethink the way we develop those AI systems for biology. Biology, which is incredibly multi-scale, where we have data on the molecular scale, phenotypic insights on the cell scale, and now we really have insights into tissue biology as well. And we need to incorporate many different data modalities, many different data types. We can leverage the hierarchy we know from biology. We know that we are gathering data on the molecular level and how they might feed into the cellular level and into the tissue level. How to get those universal embeddings across scale, across modalities, to have the most robust representation of biology that then allows us to get an understanding of what's actually going on. Ultimately, we want to understand how those cell systems change upon perturbation with a drug, upon differentiation in embryogenesis, and so on and so forth, in those dynamic processes. So we also need to build AI systems that are able to kind of forecast those cellular system changes, for example, like how a cell state changes upon perturbation with a drug. We kind of differentiate here between those universal representations that can be used and those instruments that are building on top of them that allow us now to simulate a change, to get a readout of a molecular profile, to propagate how a change on a molecular level then affects a tissue composition, and so on and so forth. And I would like to get a bit more precise, right? I will not dive into the details, but a bit of precision of what this could be, what this could mean, to give you one idea about how such a universal representation might look like, and how we build those instruments. I mentioned before, right? We have more and more pathology foundation models for RGB images, for histopathology. But how do we deal with spatial proteomics? So the images I showed you on the very first slide. What we have is diagnostic images via multiplex. So we have those images that consist of hundreds of different channels. And every channel measures the intensity or like kind of presence of a protein within a tissue. And as I mentioned, depending on the study, depending on the interest, every data set might have a different set of proteins. We are also working with clinical data. We are interested about biopsies. Data is not just falling from the sky. Usually we are working with small clinical cohorts. But in order to really understand consistent patterns, right, ChatGPT was trained basically on the entire internet. Here, we cannot just train on one single cohort. We need to gather all data sets that we can find and train on them jointly to get the most robust representation. So we have small clinical cohorts, we have a lack of consistency of labels, and every data set is going to be heterogeneous. Here, we just have 5 different clinical studies and can see which markers are measured, right? Usually we have RGB, here we have hundreds of different markers, every data set is having a different one. How do we build AI systems for this? Ultimately, what we want to build is a universal representation of those cancer tissues, like a virtual tissue that we can now query to certain downstream tasks that we are interested in. For example, clinical diagnostics, therapy selection, outcome prediction, or biological discovery task. But what we also want to build, in particular in Switzerland, is almost we call it virtual tissue database, that I can now go into and query. I have a new patient which has a surprisingly similar profile as a patient I've seen in the past. Let's look at how this patient responded, and let's use these kind of case studies to really be in we call it molecular tumor board, and influence or guide decisions that are happening between clinicians. Because now they have a toolkit that kind of lets them go into the past and find somehow cases that might be relevant and strategies that are basically data-driven. So what we have is now a system where we build those virtual representations. So we have a representation, kind of like something we can really physically grasp of our tissue. It's something that we train once, and now we can zero-shot, we call it. We have a new clinical study, we can embed it into this virtual tissue space, and now have all those different prediction and rich behavioral tasks. And we build those systems future-proof, and I will show you just in a second how. Even if you have a new marker in future, right, we need to be able to get a representation of the tissue. How do we achieve this? By really rethinking the way we design those systems, those AI architectures. By not just using a vision transformer, this is how those architectures are called, that is basically used for street view or dogs and cats, right? We need to rethink how this works for tissue biology. Meaning we need to incorporate also knowledge we have on the channels. It's not just red, it's not just green, no, it's protein ABC that's measured. And we can use protein language models, just here as an example, trained on the entire protein database that incorporate as much knowledge as we have about a particular protein. We can fuse it and find a tokenization systems for multiplex images that tokenizes the identity of a particular channel of a particular protein and its spatial location. Similar as we have tokenization systems for language, we have here tokenization systems for tissue images. And now we need to also be multi-scale in the readouts because we are interested in understanding cell identity, cell state, tissue niches that are characteristic, as well as whole sample information that we might want to predict of an entire patient. We need to also rethink the architectures and really the building blocks because now we work with 100-dimensional images. So an attention that is used in normal vision transformer by Meta, developed by Meta, is not going to scale. And also we would like to understand the difference between a spatial influence as well as which protein somewhat really guides an algorithmic decision. So disentangling them has many different effects. On the one hand, you can see here the transformer used by Recursion, big pharmaceutical company, specialized on images. They're using basically developed AI tools for many different, very different data types, not necessarily for tissue biology. You can see the moment you add a lot of different markers or dimensions, how they not scale, right? Our system scales very, very well. And because we provide meaning to what each channel means, we also benefit from it. Our performance and accuracy increases the more biological context I add. Something very obvious, right? But if I build an AI architecture that's not used to this type of data type, that's something we don't see in basically the current approaches. It's also interesting to look at this because now I can look at a tumor. Here you see a tumor. In dark you see the tumor cells. And now I want to predict cancer type. Where would a pathologist look at? Of course, the distribution of tumor cells, right? Somewhat like how they interact, how they are structured, and so on and so forth. That's exactly what we are looking at. Like here you can see the attention score. It's looking at rather the tumor regions to understand what's happening. Same, we can now zoom into certain tissue regions and understand what are the markers, the proteins that are critical for an algorithmic decision. And in the tumor regions, that's going to be tumor markers or two kind of proteins that are characteristic for tumor cells. In immune cell regions, here in red, these are going to be markers relevant for immune cells. And this is somewhat very interesting because now we can look at potential biomarkers relevant for a clinical decision because we have an understanding of what the algorithm is looking at when looking at these tumor tissues. What we're actually interested in is really looking at this from a clinical decision-making point of view. Can I now give a query tissue, for example, a tissue on the left, and find relevant cases in like somewhat our database? And as you can see here, we get 3 times an adenocarcinoma, and of course the sample we had was also 1. So you can see it matches very closely in the clinical labels. And here, I show you just as an example for 2 cases. Of course, we have all the full results that are not showing you today in the paper. But so this is really what we are envisioning. So we can now go and query most effectively our databases because we have those representations that really capture a tissue biology and allow us to now search for similar patients with a similar molecular state. Now we can go one step further, right? Like we've seen in the last month, right? This ability of those models to start correcting themselves, improving themselves, or like some form of what we call reasoning. We are not at a reasoning stage yet, but we can somewhat look at the systems built for large language model and rethink how we can use them for our models. If I have larger and larger foundation models, right, that are trained on many different technologies, do I always need the most expensive diagnostic test? Or can I map in with a cheap test because it's complemented what the model has else seen? Meaning that I can map in with whatever diagnostic test into this virtual tissue space and then use it for different downstream tasks. Can I understand what are the most important protein markers to measure? So I only need to do 1 round of cycling of imaging, but not many, right? So what are the most 5 important markers that gives me a most robust enough representation so I can do everything I want? So basically, can we have a model that understands how to select a diagnostic test in order to get the highest accuracy and kind of save cost because those tests are expensive and not feasible for every patient? And can I understand how to steer and guide experimental design? And similar as those large language models, this is based on reinforcement learning that operates on top of those foundation models. So now we are basically having an algorithm develop a policy of when to switch from one modality to the other, or when to measure which marker and what's the optimal sequence of markers to get a good enough tissue representation in order to have highest accuracy, as if I would measure the most expensive and the most accurate diagnostic test. What you see here is, in dark blue, what a pathologist would choose, like basically selected views here. That's something, I think this doesn't work. Yeah. Selected views here is basically the number of modalities or markers to measure. And you can see a pathologist, of course, knows the most important markers. You can see that the blue line, dark blue line, matches the line of the algorithm that we developed in light blue. But then after 3-4 markers, it's not so obvious anymore what's now the most informative next marker to really get this leap in accuracy, while at the same time reducing the uncertainty or increasing the confidence we have in our algorithmic decisions, something incredibly important in a clinical setting. You can see here that if we are using pathology foundation models for H&E, we are getting often an accuracy that's actually fairly good, right? If we would use only spatial proteomics in the second column, of course we are even better. We have an even higher accuracy on a prediction task. Our algorithm actually barely queries spatial proteomics. Most of the time, a simple diagnostic test is enough. But just in those critical cases, it knows when to switch. The policy knows when to actually ask and request a higher, more expensive diagnostic test. You see, we are getting the same accuracy as running always an expensive diagnostic test, while drastically minimizing the costs for a patient and for the healthcare system. And this gives you just a couple of ideas on how we are rethinking these AI systems to make them applicable for tissue data like spatial biology that's incredibly high dimensional, and how we are building systems that are adaptive to whatever is in need. One idea on how to build those systems that allow us to really somewhat forecast dynamical changes, but this we will cover very, very quickly because we are not wealthy in time. So what we really want to understand how we can forecast cellular behavior or changes in cellular functions. I have a cell, I have a measurement of a cell, and I would really understand how a cell transitions from a disease into a healthy state. And especially how does the biological features, different proteins in a cell, how do they change, right? What's really the phenotypic change of a cell? I also would like to understand how a cell changes upon perturbation with a drug. Because if I can really forecast those changes, I can simulate how a treatment would affect a patient's tumor, and then ultimately, afterwards, what's the most effective treatment to select. If I can do something like in silico, I save a lot of time, right, and reduce the number of, I kind of find the most effective treatment for a particular patient. We need to capture this on the cell level because those tumors are very heterogeneous. And I really want to understand what are the cells that are going to be responding to a drug and what are the cells that are resisting. And as I mentioned already at the very beginning, we are dealing with a situation where I don't have aligned data. I only have data before or after of a cell, one cell either unperturbed or perturbed. So instead of really operating on just a trajectory for every cell, I need to look at distribution of cell states in an unperturbed and a distribution of a cell state of perturbed cells and try to somewhat reconstruct what could have happened to explain the drug effect and learn those AI systems that can accurately forecast how a tumor responds to a drug. And one core principle, and this is of course a huge simplification of what we are going to do, are something that's known as generative AI, and I'm sure you've all heard of it. It's magical. I also used it to understand what I probably expect when I join EPFL, what would be promised, right? So we can generate from nothing and a condition exactly what's happening, right? It's kind of true, we are the lake, there are mountains, but something's missing. But so we basically do this, right? We have generative models that can basically generate from an unperturbed state of a cell the perturbed state. Meaning, given a particular drug, I can now simulate the drug effect, forecast with a neural network, a generative AI model, how those cells change upon perturbation with the drug. And even beyond, right? So that's, I said, a huge simplification. The systems we use, they actually need to be not just a random generative model, right? Because as you can see in the slide before, that's kind of what I could expect at EPFL, but then not quite the truth, right? So almost the truth, almost the truth. So what has been a work I've conducted now for many years is how to put as much kind of knowledge or guidance into the process to be as accurate as possible to really somewhat simulate cellular dynamics. We are using something that we call optimal transport that has very beautiful theoretical connections that I'm going to spare you with today, but where we are really looking at somewhat forecasting those cellular changes using maps. It has nice systems, kind of connections to dynamical systems theory where we can use partial differential equations, as well as those diffusion models here that are actually very well known in control theory or now in those stable diffusions, very powerful class of generative models. And because that has been basically the content of my PhD and someone already listened to my PhD defense, I'll spare you with the details. But we've summarized the kind of nice theoretical contribution, this new class of generative models in an ICML tutorial 2 years ago. And we also recently wrote a Nature Reviews method primer on everything optimal transport all those generative models can do for spatial omics and single cell omics. But so we're actually using those technologies in a clinical setting where we have data from the Tumor Profiler Consortium, a really great initiative within Switzerland. Here we have a rather small cohort of 37 patients for which we have samples of those patients, and we actually run a drug screen against 34 clinically approved drugs. Now we want to use those generative AI systems to forecast how every single drug would change a patient's tumor cells and then select the one that's most effective. Meaning we have a cohort for which we have both samples of cells before treatment and samples of cells after treatment, and we use those data to train those generalized perturbation models. Now for an incoming patient or a patient of interest, we can use those systems to forecast the effect of the drug. Meaning, I said it already before, right? We can look really at this feature changes, how the molecular properties of every individual cell change. And now, because we have it for a cell and its prediction, we can extract all those patterns and really capture this treatment response of a single cell. So how every single cell changed their features upon perturbation with the drug. And you will see that this is critical to really understand how effective a treatment is. Because it's a clinical study, we actually have survival data. We know what the patients actually got in a clinical setting, and we know how much longer they survived. Now let's understand how effective the treatment was. If I just look at the meta-information, like clinical treatment histories, driver mutations, number of treatments in the past, I basically get a random chance of predicting if this treatment was effective or not. Even if I look at the state of the disease of the single cell profiles before a drug, I can't really make an accurate statement on this. Even the treated state itself, right, they give a bit better idea of how effective a treatment is. But we really need to understand what has the treatment done, right? Like what has really changed? How did these single cell profiles respond to a drug? That really provides us a really high accuracy on understanding the effectivity of a drug. And then also to select the one that provides the highest, basically, effect. So these are just two examples on how we build those AI systems, right, with this overall vision of building those multimodal, multi-scale foundation models for biology to solve all those tasks simultaneously. And I mean, it might sound far in the future, and we recently had an article covering our work, which said that the eye virtual cell is a holy grail of science, but it's getting closer. And that's really what I believe, because we are in a new area of having understood how to build those generalist models, having the access to the data, and in particular in Switzerland, having access to the compute that we need to work with this amount of data that's really, really, really terabytes over terabytes of signal into a single tissue. And with this, just like one philosophical outlook, right? So I think over the last years, we fairly well understood how AI can be predictive. And I think everybody now knows, right, that AI can be generative, and we can fill in the blanks and the gaps that we might not be able to measure. But the core question is, can we query what AI knows, which we don't know, and how to do this? And then in particular, how can we reason of what is really happening? What is a driver of a disease? What is the most effective drug? And how do we alter it to make it even more effective? And now we need to think on how to do this for biological data and for biology and medicine. And with this, I would like to really thank my team, who has already worked on the very first two projects. So these are two projects that already came out of my lab. And we've just started in September, so they did a fantastic work. And I'm immensely proud to work with them. Then, of course, also my PhD advisors who set me on this path, Andreas Krause at ETH and Marco Couturi at Apple. The only time Apple so far has done work in biotechnology. So then there's my postdoc host, Aviv Regev at Genentech, and Jure Leskovic at Stanford, and many other collaborators. And then I thank also my family, and I'm happy to take some questions. Thanks so much.
Thank you. Thank you very much, Charlotte. Time for questions. Let me deliver the microphone unto you.
Thanks, Charlotte, for the great presentation. So you mentioned a cohort of 37 patients. Can you say a bit more about that? Is it just from CHU, for example? And is there a plan to have this expanded, let's say, nationwide at some point? And what are the hurdles that you did encounter? And what's also the more international perspective in terms of access to data?
Yeah. So this is indeed a super small cohort, and this is actually one of those models that has like focused on this one small cohort, and like how can we make forecasts within the cohort. What we right now actually assembled, like data sets that we assembled in the lab, they go way beyond this. So on the one hand, there's actually a lot of public data sets, and the mass, like the data scales the moment we are flexible, right? That we can combine data sets that have been independently collected. And then we get actually to patient numbers that are right now, like on our hard drives, you have, I think, 16,000 patients. But then there's also actually really, really nice initiatives within Switzerland. So there's a Swiss Personalized Oncology Initiative, where we actually also this year collect 3 new retrospective study that we are the computational team that is going to analyze them. So this is between all the university hospitals. They are basically in one single initiative that is collecting. Switzerland is very small, right? So we need to really work across university hospitals, and that's happening. And that's, I mean, I'm a biggest benefit of like many, many years of work that has been done in this space. So this is that. And then, I mean, also right now we're actually launching very new initiatives, again, all university hospitals on board, so that we actually get to patient numbers that are very nice, and in particular also to many different perspectives, like measurements of many different platforms. Maybe one last comment, right? One of the things I said is that we need to be as flexible as possible in the way we design our AI systems, because every hospital has a different technology, right? They might have acquired this technology and they might have acquired this technology. In order to gather all the data, we need to have algorithms that can deal with diversity among them. And that's exactly what we're up to. So we shouldn't ask everyone to have the same system. We should rather develop algorithms that can also make use of everything that has been historically collected. And we have these atlases. I didn't give you the numbers, but we had millions and millions of cell profiles that we have measured. And if I'm flexible enough and I can incorporate them all into the training of our systems, then I think that then we actually unlock useful signals that we can use that are task agnostic and can be used for many different type of downstream tasks.
Thank you very much.